Welcome back to our channel, My BioNotes. So, in this video, we'll see about the meiotic type of cell division. So, before going into the video, please subscribe our channel and also click the bell button for getting more updates from our channel. So, come let's see about this meiotic type of cell division. So yeah, it's a cell division only, but here it is a special kind of cell division, and here the chromosome number, it gets half. So you all know we have 46 chromosome, 23 pairs of chromosome, it gets half in this case. So after this meiotic type of cell division, you will be getting 4 haploid cells. So it's the haploid phase in sexual organism. This meiosis is the haploid phase, and the diploid nature is restored by the fertilization. So after the fertilization of the cells again gets diploid. The meiosis is seen as gametogenesis in case of plants and animals. So plants and animals meiosis occurs as gametogenesis. And here in meiosis you have 2 sequential cycles of nuclear and cell division. So the nucleus and the cell gets divided like in mitosis. So here in meiosis you have meiosis 1 and meiosis 2. So here in meiosis 1, like in mitosis, we have prophase 1, metaphase 1, anaphase 1 and telophase 1. And meiosis 2 is also divided as prophase 2, metaphase 2, anaphase 2 and telophase 2. And there is a single cycle of DNA replication. Of course, the DNA must get replicated because after the cell division, the cell must have a DNA, right? So there is a single cycle of DNA replication. So in prophase 1, we have different stages like the leptotene, zygotene, PACHYTENE, DIPLOTENE and DIAKINESIS. So this prophase 1 of meiosis 1 is a longer and more complex phase of this cell division. And coming to the leptotene stage. So what leptotene means in Greek, if you see, means leptotene means thin threads. So obviously it refers to the chromosomes. So it appears as thin threads. So when you view it under a light microscope, the chromosomes, they gradually become visible during this stage of leptotene. And there is compaction of chromosome. So we have seen in mitosis there is condensation. We can call it as a thickening or a compaction of the chromosomes. So it gets thickened. And coming to the next stage, that is the zygotene. So zygotene, it's a Greek word, of course. From the word itself you can infer. So the zygotene stage in Greek, which means paired threads. So the threads are getting paired. Here the thread refers to the chromosomes. So here in zygotene stage, the synapsis occurs. So what is a synapsis? The process of association in which the chromosomes start pairing together. So here the chromosome pairing occurs, which we call it as a synapsis, which is a connection or a junction. You call it as a synapsis. Coming to the zygotene, in zygotene, there are homologous chromosomes. So the paired chromosomes, you call it as a homologous chromosomes. So if you see in an electron micrograph, you will see a chromosome synapsis and also a synaptonymal complex. So as you can see in the picture, so this is the synaptonymal complex. You can see the 2 homologous chromosomes having the centromere and the kinetochore with the sister chromatids. So they form a complex, which we call it as a synaptonymal complex. So what you call as a bivalent or a tetrad it is a complex formed by a pair of synapse to homologous chromosome that you call it as a bivalent or tetrad. So this is about zygotene stage. So coming to the next stage, which is the PACHYTENE. So what occurs here? What PACHYTENE means na? It means thick band. So you can see thick band in this stage. So here 4 chromatids of bivalent chromosomes. So the chromosome is bivalent, 2 chromosomes are there, but the chromatids are 4. They become distinct and they are visible as tetrad since it's 4 chromatids are there. And here comes the most important event, which is crossing over. So this what makes meiotic division different from the mitotic division. So what happens in this process of crossing over, if you see, means there is exchange of genetic material between 2 homologous chromosomes. So that's what occurs. It's, see here, the homologous pair after synapsis, they cross over to get the recombinant chromatids. So see how they have recombined. So there has occurred a exchange of genetic material. So for this only the synapsis and the connection has occurred for this event of crossing over. Next, coming to the recombination nodules. So what you call as recombination nodules means the swelling or a lump you call as nodule. So in this case, recombination nodule. So here you can see recombination nodule at the site at which crossing over occurs between non-sister chromatids of homologous chromosome. There you can see the recombination nodules. So here, and the chromosome, no, they are linked at the site of crossing over. So they remain, and the crossing over site, they remain linked. And coming to the diplotene stage, here there is dissolution of the synaptonemal complex. So now there is a breakdown because the crossing over has occurred. Now the synapses has to be broken. Everything comes to an end like this. After crossing over, now all get dissoluted because crossing over is the main name. Crossing over occur, now there is dissolution. Next, separation of the recombined homologous chromosome of the bivalent chromosome, and except at the site of crossover they get separated. And chiasmata, what you call as chiasmata, is the X-shaped structure. So you can see a X-shaped structure here of the homologous chromosome, that you call it as a chiasmata. And in case of oocytes of vertebrates, this diplotene stage can take months to years to complete. So it's a big duration in case of oocytes of vertebrates. So here you can see a clear picture of the leptotene, zygotene, PACHYTENE and diplotene stage, and you can see how the 2 homologous pair of chromosome they get associated with each other with the sister chromatid just for the crossing over to occur. So let's not go detail into that. Next, coming to the stage of diakinesis. Di means 2. So here it appears as double band in case of diakinesis. Here there is termination of chiasmata. So here this is the end, and the chromosome is fully condensed in this case, and here the meiotic spindle gets assembled. So mitosis means meiotic spindle. Here since it's meiosis, it's called meiotic spindle. And there is separation of homologous chromosome. So the homologous chromosome tend to get separated. At the end of diakinesis, the nucleolus disappears. The nuclear envelope, no, it breaks down. and there is transition to metaphase. So from diakinesis, the next stage, metaphase, occurs. So thank you all for watching my video. என் வீடியோ உங்களுக்கு பிடிச்சிருந்தா லைக் பண்ணுங்கள், ஷேர் பண்ணுங்கள், and also bell button click பண்ணுங்கள். அப்போ தான் நாங்கள் ஏதாவது வீடியோ போட்டால் உங்களுக்கு நோட்டிஃபிகேஷனில் வரும். Happy learning to you all. Thank you.